We just saw the cellular and molecular basis of olfaction, our ability to smell. Now let's see the cellular and molecular basis of vision, how we are able to see. So again, as I mentioned, it's G protein coupled receptors that allow us to do it. And a very similar receptor is also present in bacteria that allows it to make ATP. So Halobacterium salinarium basically has a specific type of receptor, G protein coupled receptor, and this receptor is covalently attached to another molecule which is 11-cis retinal. This molecule is covalently attached to this receptor. When a photon, a light particle, hits this retinal, it has a conformational change. This conformational change allows one proton molecule to be ejected outside the cell. It creates a proton gradient. Your biochemistry teacher will tell you how a proton gradient can be used to generate ATP. For us, on the other hand, we use the same system to be able to see. We have two types of cells, rod cells and cone cells in our eye. We are going to talk about the rod cells, which are responsible for black and white vision. So in these cells, there are stacks, there are membranous enclosed little sacs. These sacs have specific proteins, G protein coupled receptors, which have the similar structure to that bacterial receptor. This receptor is called rhodopsin in this case. This receptor, when it receives a photon of light, this photon is absorbed by the same molecule, the retinal. It goes through a conformational change and it activates a G protein. This G protein, as you know, will get rid of GDP and attach a GTP molecule instead of a GDP molecule. This alpha subunit, when it dissociates, it will go and activate a protein called phosphodiesterase. This phosphodiesterase will basically, as the name suggests, it will convert the cyclic GMP into a linear molecule of GMP. So this is our phosphodiesterase. In response to a photon of light, G protein was activated. Alpha subunit of that G protein activated phosphodiesterase. This phosphodiesterase will hydrolyze GTP molecules into cyclic GMP molecules into non-cyclic GMP molecules. Cyclic GMP molecules, which are present in absence of light, attached to an ion channel receptor. This ion channel receptor basically causes the cell to be depolarized. So once this happens, the cyclic GMP, once the phosphodiesterase gets rid of cyclic GMP, this ion channel will close, and now the neuron, rather than becoming depolarized, will become hyperpolarized. This hyperpolarized neuron, basically this neuron is an inhibitory neuron. Once this neuron is hyperpolarized, its inhibitory effect will be limited. So this is again the structure of our rod cell. You can see these sacs which have this rhodopsin molecule on them. When light hits them, the G protein activates the molecule that is going to degrade the phosphodiesterase molecule, which is going to degrade cyclic GMP into linear GMP molecule. That now the signal has been transmitted from this region of the cell to the membrane, because once the cyclic GMP is degraded, it will no longer be able to open those ion channels. When ion channels are closed, the neuron will become hyperpolarized and it will not be able to inhibit. When this inhibition is restricted, the signal is sent to the neuron and to the brain, and it can create one image. The brain will detect that one photon. So here now let's look at it in a different way. One rhodopsin molecule absorbs one photon. The rhodopsin molecule is basically the G protein coupled receptor. In this case, we are calling it rhodopsin. This one rhodopsin molecule activates 500 transducin molecules. Transducin molecule is basically the G protein. The G protein in our rod cells is called or referred to as transducin. One rhodopsin results in activation of 500 transducin molecules. These 500 molecules activate one phosphodiesterase molecule, which results in production or hydrolysis of 100,000 cyclic GMP molecules and results in closure of 250 sodium channels. The ultimate result is the rod cell is hyperpolarized by 1 millivolt. Removal of this inhibition sends the signal to brain that a photon has been detected by such and such rod cell, and once the brain receives this information from many hundreds of thousands of these rod cells, the brain is able to construct an image which allows us to see. I would like to recap again that some of the G proteins we have talked about. GS is the stimulatory G protein which activates the adenylyl cyclase once it interacts with the receptor which has been bound to its ligand. G olfaction, this is the G protein which is present in the neurons of our nose. GI is inhibitory G protein which inhibits the function of adenylyl cyclase. GT is G transducin. It is a G protein which is present in our eye rod cells. GQ, the alpha subunit of this G protein, basically activates the phospholipase C, which results in production of DAG diacylglycerol and inositol phosphate, triphosphate. So we have seen different types of G proteins that play a specific role in different types of cells. Some of them activate adenylyl cyclase, other inhibit adenylyl cyclase, some activate a different type of enzyme called phospholipase C. In all, these G proteins help us see and smell. So an important signaling system that we have evolved over the course of evolution.